Welcome everyone to section 2 on cell division, and we're going to focus on mitosis in this video. So when we talked about the cell cycle, remember the cell cycle consists of G1, then S, G2, followed by M. Of this cycle, only a small portion is M phase, about 10%. Of the entire cell cycle is mitosis, which is what this entire section is about. But the cell cycle, most of from here all the way to here is interphase. Okay, and that's G1, S, G2, where the cell grows, the DNA replicates, prepares for division, and then it actually divides during mitosis. So we've already talked about interphase, but now we're going to go through the actual M phase or the mitosis phase. So here's our new class pet. Our puppy's name is Mitosis, and Mitosis is still a young puppy, and she needs a pee mat. Okay, she needs a pee mat so she doesn't pee all over our carpet. So that's kind of an acronym that we're going to use to kind of remember the order of mitosis for prophase, metaphase, anaphase, telophase. Okay, or pass me a taco. If you want to remember it that way, you can remember it that way. But we're often going to refer to our puppy Mitosis, who needs a pee mat because she's still young. So a very small portion is actually this division stage. I had just said 10%. This says 4%. It's a small percentage. Okay, but the 4 steps are prophase, metaphase, anaphase, telophase. And then at the very end, we have this step called cytokinesis, which in your POGL activity, cyto means cell, kinesis means the cutting or splitting of. So therefore, that's just kind of that last step. That's this part right here where we're pinching that cytoplasm. A lot of times we just kind of call it part of telophase, but technically it's not mitosis. So some important terms that you need to use, and I know you've seen a few of these terms already, but we want to keep emphasizing them because they're important, because they're terminologies we're going to use. DNA. This is all about dividing DNA when we talk about mitosis. So we know DNA is in the nucleus, and most of the time the DNA is something called chromatin form, and chromatin is uncoiled. It's like this bowl of spaghetti, and this was in the last video, I know, but we really want to emphasize this. So most of the cell's life, because most of the cell's life is in interphase, it's in this chromatin, uncoiled, all stretched out, barely visible within the nucleus. However, some important terms that when we talk about cell replication, that chromatin condenses to form something called chromatids, and chromosomes, and sister chromatids, and centromeres, and all that stuff. So these are some terms. We kind of drew this picture, started drawing this picture on the screen in class the other day. But when we talk about this whole entire structure, so part A, this entire structure is a chromosome. The entire thing is a chromosome. Okay, and 1/2 of the chromosome is called a sister chromatid. So it says either C or D. So notice how we kind of color coded. So the yellow part right here is a sister chromatid to the blue part, which is the other sister chromatid. They are sister chromatids to one another. In this point, their DNA is coiled or condensed, and the 2 halves are genetically identical to each other. So this is the result of S phase. During S phase, the DNA replicates, and now we have a copy on this side, and let me rephrase that. We have one copy of DNA on this side, and then the other copy is on that side. They're genetically identical to one another. This little pinch point right here is referred to as the centromere. And then we talk about these bands, and these bands are these sections of DNA or a location of DNA that are coded for a particular trait or a particular gene. And a gene is a location, and some examples of a gene would be hair color or eye color. Notice how I didn't say blue eyes or brown eyes. I said eye color. I said hair color. But these are genetically identical to one another. So the gene here is the exact same gene as here. So like this would be blue eyes, and this would also be blue eyes. Okay, but genes are the trait. So we also talk about things called homologous chromosomes, and that will be coming as we kind of progress here when we get to meiosis. But homologous chromosomes, you are a result of sexual reproduction. One of these chromosomes, we have 23 pairs of chromosomes. The green chromosomes, we say, came from mom, and these red chromosomes came from dad when you were formed. Combination of egg and sperm cell. These are homologous chromosomes to one another. One came from your mom, one came from your dad. This is a different pair of homologous chromosomes. They're different sizes. One of these came from mom, one of these came from dad. Okay, so we talk about these two are homologous chromosomes. These two are a different set of homologous chromosomes. Homologous chromosomes are different colors. One came from mom, one came from dad. They have the same shape and the same size. They carry the same genes, but different possibly forms of those genes. So when we talk about genes, again, we're talking about hair color. So this right here is maybe hair color. So this is mom's hair color. This is mom's hair color because these are sister chromatids, genetically identical. So this is maybe blonde hair and blonde hair. And then this is also hair color over here, but now this is dad's hair color. So this is brown hair, and then this is brown hair. So they may have a different form of the allele, and we'll get to alleles in a little bit. Sister chromatids are genetically identical. This half is identical to this half. This half is identical to this half. But these are not identical to one another. So the first step of our PMAT acronym is prophase. So prophase is the first step, and it comes after G2. And there's some key events that we need you to know when we talk about prophase. So prophase, the chromatin, which is our uncoiled DNA, condenses into chromosome or chromatid form, we sometimes say. So we start to see these X-shaped structures starting to form. Okay, so we now start drawing them as like chromosomes or these X-shaped structures. During prophase, the nuclear membrane, the nucleus, disappears. That's why they have these dashed lines just to show like, hey, it's dissolving, it's disappearing. And we start to get these things called spindle fibers starting to form. And they're going to be ropes that are going to be pulling on these chromosomes in a little bit. So that's prophase. Prophase condenses, the nucleus dissolves. Meta. I often remember meta and middle. Meta and middle. So metaphase, middle. What goes to the middle? The chromosomes go to the middle. The chromosomes all line up in the middle. And the reason is these spindle fibers are attaching. We have all these spindle fibers that are attaching to that. Okay, so metaphase, the chromosomes line up in the middle. Chromosomes in the middle. During anaphase, remember anaphase apart. Anaphase apart. So when we talk about anaphase, we had this chromosome here, but now what happened is it split apart. We had another chromosome, but now it's split apart. The sister chromatids pull apart. Okay, so what pulls apart? The sister chromatids pull apart. Then so remember, PMAT, prophase, metaphase, anaphase, now telophase, and then again, we often group cytokinesis together. Telophase is when the nucleus starts to reform. So we get this nucleus starting to reform here. A lot of times we kind of do this dotted dashed line, kind of like when it was dissolving, now it's reforming. And then the DNA starts to uncoil. So here's our uncoiled chromatin, uncoiled DNA. That's the telophase part. And then cytokinesis is this splitting of the cytoplasm. Important is, as a result, let me change colors to try to show that a little bit. This results in I thought I just changed that color. Sorry. 2 genetically identical diploid cells. 2 genetically identical diploid cells. And we'll get into diploid and haploid next section. 2 genetically diploid cells. This cell is genetically identical to this cell. So mitosis, big picture. We start with 1 cell, we end up with 2 cells. They call it a mother cell, creates 2 daughter cells. So 2 daughter cells are come from one parent cell. The daughter cells are genetically identical to the parent. This cell right here is genetically identical to these two cells over here. Cells that are created for mitosis are made to help the organisms grow, repair, replace. Grow, repair, replace. Some organisms also go through and they use this as the other reason why a cell will divide, to reproduce, and they would reproduce asexually. So that's mitosis. If you have any questions, please let us know.